Diagnosing autism from only a single strand of hair? Believe it or not, this is a real area of cutting-edge scientific research. But is it really possible? Stay tuned to find out. Hey there, welcome to Science is You. We're a young but growing channel with the mission to bring you reports of real biotechnology research directly from the source and make scientific research accessible and fun for everyone. To support us, please subscribe and share with your friends. Thanks for watching. Autism spectrum disorder is a neurodevelopmental condition that can cause a range of behavioral differences. This can include impacting ability to communicate and interact with people, altered sensitivity and repetitive behaviors, and for some people potentially impair areas of life like work and social activities. Also, autism commonly presents alongside other conditions like ADHD. The World Health Organization reports around 1 in 100 children to have autism, but CDC estimates are a bit higher, around 2 to 3%, with differences in demographics and region impacting prevalence. The average age of autism diagnosis in the US is around 4.5 years, which is pretty young, but these first few years of a child's life are a critical window of neurological development. According to the CDC, 85% of children diagnosed with autism already had records of concerns about their neurological development. A challenge facing autism therapy is the development of interventions that can prevent these developmental delays. But there's a lack of diagnostic tools and biomarkers or biological indicators of disease that can help diagnose autism even earlier in life, in infancy. In this scientific study, the researchers experimented with using a single hair to diagnose autism. Here's what they did. In this study, the researchers used a cutting-edge experimental technique called laser ablation to examine molecules contained within a single hair. This technique uses a powerful laser to remove small amounts of material from a sample. A laser is scanned along the length of the hair in this case, which creates a puff of particles. These particles were then analyzed using mass spectrometry. Mass spectrometry is a method of sorting a mixture of molecules based on size or mass and their electric charge. In this way, an unknown sample mixture can be broken down into its individual components and the identity of the molecules that make up the sample determined. But what exactly were they looking for? Exposures to toxins like certain metals and nutritional deficiencies as a fetus or in infancy have been previously linked with increased chance of developing autism. Looking just at an individual's genetics could miss these factors. The researchers reference a previous study they did that examined teeth, where they believed to identify some patterns that were different in individuals with autism. But of course, using this approach to aid early diagnostics isn't really ideal since teeth aren't really accessible in infants and very young children. Hair, on the other hand, is easily accessible in babies. Snipping a strand of hair would be even less invasive and painless than, say, drawing blood. Not only that, but hair strands grow at a rate of around 1 centimeter per month. Another way to think about this is that each centimeter you travel down a hair strand gives you a history of the previous months. So by scanning the laser down the hair strand and analyzing chemical composition, the researchers aim to get an idea of possible past prior exposures or deficiencies over time, not just a snapshot of one moment. But whose hair did they study? Participants came from 3 regions located around the globe: Japan, Sweden, and New York. Participants in Japan were already part of the larger Japan Environment and Children Study, which was an investigation of environmental factors that might impact child development and health. As part of the study, specimens, including hair, were taken from babies around 1 month old, and the children were followed up after a few years to determine whether or not a diagnosis of autism was present. Out of a total of over 80,000 people who were part of this study, the researchers randomly selected 220 participants, some who had been diagnosed with autism and some who were not. In Sweden, pairs of twins had previously been recruited for a study of autism and ADHD, with an average age of around 14 years. Similarly, the researchers contacted parents of siblings where one child had autism and another did not, who are participating in ongoing studies at Mount Sinai Hospital in New York, with an average age of 5 years. Here's their final sample count. They are able to access hairs from children of a wide range of ages and from different geographic regions, but there's also some deeper reasoning behind the groups they chose. The Japan-based study collected hair prior to the onset of autism in infancy, whereas examining twins or siblings where only one was autistic could account for some sort of underlying genetics that might be influencing hair composition other than autism. So they scanned the hairs down a length of around a centimeter, which would correspond to the past month of growth. And what did they find? They found over 500 features of the hair composition that were statistically different between individuals with autism and those without. This was based on patterns of metals and other elements that were present in the hair. But how to take this huge data set and turn it into an actual diagnostic tool? The researchers here applied machine learning to identify patterns in the composition of hair that were associated with autism. Machine learning involves feeding a computer sets of data that are associated with something like a disease or a condition and other data sets associated with healthy or controlled individuals. These are called training sets. There are different algorithms or instructions we can give to the computer to cause it to identify patterns in the data that would otherwise be way too complicated for us to recognize just by eye. This topic can get very complicated, but to envision the process, imagine we are trying to teach someone the difference between a dog and a cat. Trying to describe a cat versus a dog might be kind of difficult in words. They both have fur, a tail, maybe pointy ears, etc. Many of us can easily recognize this, of course, but it's really based on experience. If we see lots of different pictures of cats and dogs, we can be trained to recognize small differences, maybe ear shape, facial expressions, posture, and the more examples we see, the greater the likelihood that we can accurately identify an unknown animal in the future. When talking about the ability of a computer program or model to predict a condition or disease, there are two criteria that are especially important: sensitivity and specificity. Sensitivity is the ability to detect true positives, or in other words, if you have a condition, the probability that the test will pick it up. Specificity is the ability to detect true negatives, or in other words, not to say you have a disease or condition if you don't. So it's both of these things together that describe how likely the test is of being right in your diagnosis. Ideally, we would want both the sensitivity and specificity of the test to be as close to 100% as possible. But of course, in reality, we wouldn't expect a test to always be 100% accurate. Pretty much every test can produce false negatives and false positives. The overall accuracy of a test is basically the percentage of correct predictions. So what was the accuracy of the model the authors of this study built to diagnose autism from hair? The researchers stated that their overall accuracy was 81%. The sensitivity or true positive rate of the test was 96%, and specificity or true negative rate was 75%. So this implies that when the test was wrong, it leaned towards saying someone had autism when they didn't. So when can we expect to see this hair test in the clinic? So there are some limitations of this study. Although the study took into account different populations around the globe and involved several hundred participants, this is still a teeny tiny fraction of the global population and doesn't necessarily reflect the diverse demographics of many regions and ethnicities. Future studies are needed to see if this model holds up in even larger, more diverse populations. Also, another limitation the authors bring up is that they only used one hair in these tests. They suggest accuracy might be improved, for example, by looking at a few hairs instead of just one, but this wasn't yet tested here. Another huge consideration the researchers bring up is that autism is on a spectrum of symptoms. This test was based on a yes-no answer, but with more data training sets and groups of people, perhaps such a hair test could be made to function on more of a rating scale. Also, what about the ethical issues of a tool that diagnoses a neurodevelopmental disorder before the emergence of symptoms? Certainly, this would be beneficial to some people, enabling early interventions and possibly prevent developmental delays or challenges later in childhood. But what about false negatives and inaccurate diagnoses, or even just in general? Could such a diagnostic perhaps fast-track people into treatments or therapies they don't need nor would want? The scientists here do stress that the test they are proposing here should be viewed as a diagnostic aid that can assist in the early detection of autism, but must be in conjunction with additional clinical evaluations. What do you think? Share in the comments. What if the accuracy of the test were even higher, or could tell where you or your child falls on the autism spectrum? Would that change your opinion one way or the other about the test? If you found this video interesting, don't forget to subscribe and share to help out our mission of spreading awareness of real biotechnology research. And remember,